Hello, my name is Margit Tolle. Today I'm presenting a presentation titled The Effect of Silicon on the Organically Grown Leaf Celery Transplants Growth and Quality. This presentation has been presented at IPNC 2022 conference in Brazil. Short introduction. Silicon is the second most abundant element after oxygen in soil. Silicon dioxide comprises 50 to 70% of soil mass. All plants rooting in soil contain some silicon in their cells and tissues. Supplying crops with adequate plant available silicon can suppress plant disease, reduce insect attack, improve environmental stresses tolerance, and increase crop productivity. Silicon plays important roles in mitigating the biotic and abiotic stresses. Biotic stresses are insects, pests, and pathogens, and abiotic stresses are metal, salinity, drought, chilling, freezing. SAAT. The application of stabilized silicic acid is called silicic acid agrotechnology, which means SAAT. This technology has shown to be very effective on almost every crop. Increase of root system, longer stem and tillers, increase of leaves resulting in more yield and higher quality. SAAT decreases as well biotic and abiotic stresses, and therefore you can apply a lower rate of pesticides, which at least 50%, and you get lower infection rate, and the product is safe as well for the plant, the soil, the farmer as the consumer, and ecologically friendly. Purposes. The problem. No information about silicon influence on leaf celery transplants, growth and nutrient content. Aims. The purpose of this investigation was to assess the influence of silicon on the growth and nutrient content of leaf celery transplants. Another aim was to see whether there is an effect of silicon so early in plant development. Materials and methods. Experimental site and time. Experimental site was Estonian Crop Research Institute and their greenhouses, and time was winter of 2014. Now I make a little remark that I'm not working at Estonian Crop Research Institute anymore, I'm working at NPO veggies cultivation. Substrate. Seedlings and transplants were grown in Novarbo B2 organic biolan substrate for organic cultivation. The pH of silicon treatment and control solutions were 5.5. The lighting at plant level 12,000 lux. Day and night temperatures were 20 and 18 degrees of Celsius respectively. Replications and experimental design. Randomized block design, plot size 6 plants, 4 replication and experiment repeated 2 times. Results collection. We collected the height and stem diameter measurements and laboratory analysis, nitrates, nitrogen, phosphorus, potassium, calcium, magnesium content. Statistics was done by ANOVA Excel 2010 and comparison of means by Fisher's LSD test. Treatments. We had 2 treatments, silicon treatment and control treatment, and in silicon treatment, the plants were sprayed with silicic acid 2 milliliter per liter solution. First time, first through leaf stage. Second time 2 weeks later, and third time again 2 weeks later. This silicic acid 2 milliliter per liter solution was sprayed to the plants in silicic acid treatment, while in control treatment the plants were sprayed with water at the same time frame. Results. The height of leaf celery transplants treated with silicic acid. You can see here that the height of transplants was higher in silicic acid treatment compared to control treatment. The stem diameter of leaf celery transplant treated with silicic acid, you can see here that the stem diameter was increased in silicic acid treatment compared to control treatment. The content of nitrates in raw leaf celery transplants and the content of nitrogen, phosphorus, potassium, calcium, magnesium, and in leaf celery transplants dry matter. You can see here that the nitrates content was not significant in leaf celery transplants, while nitrogen, calcium and magnesium contents were higher in silicic acid treatment compared to control treatment, and phosphorus and potassium didn't show statistically different results. Why is we all know that nitrates can be in big concentration harmful for plant growth, and phosphorus is needed for root growth, and potassium is needed for stomatal opening and water relations in plants, and calcium is making plants stronger against pests and diseases, and it's not mobile in plants. It goes through the plant with water flow of transpiration, and therefore it's very useful to get increased calcium content, and it means that cell walls are stronger and pests and diseases cannot invade the leaf. And magnesium is also important in plant protection. With higher magnesium content you also get less pests and diseases on plants. Picture of leaf celery plants in the experiment: on the left hand side seed treated plant, and on the right hand side control plant. And you can see that the left hand side plant is more flushy than the plant on right hand side. Conclusions. Silicic acid improves the quality of leaf celery transplants. Leaf celery transplants were taller and stem diameter was greater in silicic acid treatment compared to control treatment. The content of nitrates in raw leaf celery transplants was not statistically different between treatments. The content of nitrogen, calcium, magnesium in leaf celery transplants dry matter were higher in silicic acid treatment compared to control treatment. The content of phosphorus and potassium in leaf celery transplants dry matter were not significantly different between treatments. And overall conclusion can be that silicic acid treatment can be used by production of leaf celery transplants, making the transplants taller and with greater stem diameter, and transplants contain also more nitrogen, calcium and magnesium. And here you can see that I published a book about silicon in 2020, silicon as a challenge for sustainable vegetable crop production, and you can buy it from www.vegiscultivation.com/books. And this book contains 6 different scientific articles and shows how the growth, yield and nutrient content of vegetables in crop production can be influenced by silicon treatment. You can find there a silicon review article, silicon and tomato transplants, silicon and cucumber transplants, silicon and iceberg lettuce, silicon and leaf lettuce, silicon and field pea articles. I would also love to acknowledge Rexil Agro PV who supported me by getting travel grant to attend IPNC 2022 conference, and also for the SAAT training. And I would like to acknowledge the Estonian Crop Research Institute and PRIA in Estonia for financing this research. Thank you very much.